Hi, this is Mrs. Alexander, and today I'm going to go over the front load lesson for activity 231. It's all about osmosis. And osmosis relates to diabetes and diabetics because usually their blood is at a constant high level of sugar. And the osmosis is the process by which water is trying to regulate or balance out the different solutes in the blood and the system. So osmosis is a form of diffusion, and diffusion is whenever molecules spread out over a large area, and we give a specific name for when water does this, and we call that osmosis. Your body has semi-permeable membranes, which are membranes like your skin and your phospholipid bilayers in which cells allow certain things to enter and leave, like water, sugar, salts, things like that. A vocabulary term you need to be familiar with is a concentration gradient. A concentration gradient is any sort of pressure or area of high or low concentration that molecules or particles move from one area to another. Think of it like a ramp or a slope. Concentration gradient, it goes from high to low if we're talking about diffusion and osmosis. High to low, think of it going down a hill or down a river. And so whenever there's a high amount of sugar, it wants to go, water wants to move that sugar and balance it out so that there's an equal amount on both sides of the gradient. Molecules are always moving. They're moving randomly around. They're bumping into one another, and they're trying to reach equilibrium or equal amounts on different types of semi-permeable membranes. When this happens with particles like salts and sugars, we call that diffusion because it's the movement of molecules. But when it happens with water, we call it osmosis. Notice from T1 through T3, that just kind of shows you how a concentrated area of particles is spreading out and moving further and further apart. There are some factors that affect the rate in which things can diffuse or separate and become equal. The size of the molecule, so how big or small it is, the size of the pores in the membrane, the temperature, the pressure, and the concentration. The rule of thumb for temperature, pressure, and concentration is that the larger or the higher, the faster it will move. So if we heat up something, it makes those molecules go faster. If we put it under more pressure, it makes it go faster. Or if we put a lot of an enzyme, it will make something go a lot faster. And then the size of the pores or the molecule just really depends, kind of like doorways. If something's small enough to go through the pore or the channel, then it can just diffuse on through. But if not, and it needs help to be carried through, or energy needs to be required, then we actually have to do something called active transport. Note that diffusion and osmosis, it does it all by itself. It goes down a concentration gradient, and we call that passive transport. That's when it passively does it and no energy is required. So here's a picture of equilibrium. Again, you start off with a solute or a substance, and it's trying to diffuse or move across a membrane and spread out equally. Solutes are the particles like sugars and salts. Solvent is the water portion of that solution. A good little rhyme to remember is that solutes suck. Solutes will suck water towards them, and they will move around, and they will try to pair up with a water molecule, a free water molecule, so that they can become diluted. Again, the definition of osmosis is the movement of water across a membrane. Note, here's a little picture of a beaker with a line down the middle. That line's representing some sort of membrane. Think of it like a plastic bag or your skin or a phospholipid bilayer, and you've got your large molecules and you've got your small molecules, and they will move back and forth until you can reach an equal concentration. Notice the right-hand side in the first picture has a lot more solutes, a lot more red dots or sugars. Sugars will suck the water towards them. That's why in the picture on the right there's less water on the left side of the beaker, because the water has been sucked to the right. Solutes suck. Here's another picture of the same situation showing the direction or movement of water. It's the water that moves, not the particles, because remember, the solutes suck the water towards them. We're going to be doing this in class. We're going to set up an experiment where we're going to put baggies into beakers, and the baggies are going to have solutes inside, and the beaker is going to have solutes inside. But we're going to set them up differently so that sometimes the concentration is higher inside of the baggie, other times it's higher outside of the baggie. And you guys will have to determine which area has a higher concentration of solutes like sugars and salts versus the other. Remember, solutes suck, so that'll help you figure it out. Small particles or molecules like water can pass easily through without any energy, called osmosis, but larger molecules, complex carbs, they actually have to use channels or pores. Think about a glucose transporter or an insulin receptor. Those things are actually proteins embedded in the phospholipid bilayer, or proteins embedded into the cell membrane. They have to open and close, and they're selectively permeable, meaning they select what can enter and leave at certain times. They have to have different signals from the insulin to open it up to allow glucose to get in. So it's a lot more than just simple diffusion. Active transport is required as well. Okay, in basic biology or pre-AP or whatever you might be in right now, you've probably talked about the words hypo and hypertonic. Remember, hypo means low, hyper means high, and when we say tonic, we're talking about the amount of dissolved particles. So we've got a cell in red. This is representing like a red blood cell, and we've got it in a beaker full of water. In the first picture, water is going into the cell, making it swell. Because remember, solutes suck. So the solutes inside of the cell are sucking the water towards it, making it get bigger. I think of hypotonic solutions cause the cells to look like hippos, really big and fat because they're swelling. Unlike on the right, we have a hypertonic solution. We've got a red blood cell, and there was a lot of sugar or salt, some sort of particle around the cell in the beaker already. It would be like taking a cell and putting it in a beaker of salt water. And the solutes that were around the cell sucked the water out of the cell and caused it to shrivel up and shrink. My little rhyme is I think of like really hyper people always moving and becoming active and sweating a lot and shrinking. They're smaller than people who are less active. So I think hypo, hippo, hyper, skinny, small, shrinking. It kind of helps you with the visual. We have one more type, and that's called isotonic, and the prefix iso actually means equal. And all that means is that it's already at an equilibrium point, so the amount of particles inside and outside of the cell are equal. So water still moves in isotonic solutions. It still goes back and forth, but at an equal rate. So you don't really see a net change or a change in mass of the cell or of the water, because solutes are constantly sucking water into the cell, and the solutes that are around the cell are constantly sucking water out of the cell. So it's getting sucked in and out. Water is moving back and forth. Here's a picture that summarizes all three. Shows blood cells, important because when diabetics have blood cells that are in a constant state of hypertonic solution, a constant state of really sugary water, it's causing our cells to shrink whenever you have diabetes. And that means that your lips and your skin and your taste buds, you're constantly thirsty because your skin in your mouth is always shriveling up. It's really dry because that hypertonic solution is causing the cells to shrink. So you're wanting to put more and more water in there so that you can kind of balance it out. This is also really important with plants, especially the type of plants. There are some underwater plants that only are suited to live in ocean water. It's really salty. And then there's some freshwater plants that cannot take the salty water. It all goes back to that hypertonic or hypotonic solutions. It doesn't matter if you're an animal cell, a blood cell, talk about diabetes or plants, it is still going to have the same result. Hypotonic solutions will cause cells to swell. Hypertonic solutions will cause cells to shrink. Here's a microscopic view of that happening. Elodea is the name of a plant that's an underwater plant, and it shows the little green chloroplasts. It shows the square-shaped cell wall. The picture on the right is all shriveled up and shrunken, and most of the chloroplasts are all in a ball in the middle, versus the one on the left where it's kind of plump and they're all more spread out. Again, we were just talking about hypertonic and hypotonic, and we're talking about something known as passive transport. Again, passive transport does not need any energy. It's the movement of particles across the membrane from high to low. High meaning a lot of that solute, low meaning where there's not very much, because solutes suck water towards them. If you have any questions in class, let me know. Where we're going to go from here is we're going to set up our lab in class to look at what it's like for the cells inside of diabetics to swell and shrink. So here's the picture of the lab that we're going to set up in class, in which we're going to place a baggie full of material, something like a salt sugar mix with water, and we're going to put that bag in a high concentration of salt or sugar. We're going to put a separate one in a bag of isotonic or no salt or sugar, equal, and then we're going to have one that's in a very hypo or low amount of solutes. And over time we should be able to see that bag shrink or swell just like the cells did in the pictures I just showed you. How does this relate to Anna Garcia and our diabetes that we've been learning about? Well, diabetics aren't always just hypertonic solution style blood. Sometimes they go through highs and sometimes they go through lows. We call it hyperglycemia and hypoglycemia. Just like the words hypotonic and hypertonic, but instead we're saying glycemia, meaning their blood sugar levels. A diabetic has to constantly check their A1C levels, and those A1C levels are used for type 1 and type 2 diabetics, and it gauges how well they're maintaining their diabetes. A normal level for diabetics would be an A1C level between 4.5 and 6%. If you don't have diabetes, but you're kind of on the cusp or you need to start monitoring yourself, that's when you have between a 5.7 and 6.4% A1C. However, whenever you're uncontrolled diabetics, usually see an A1C level of 8% or higher. Here's another picture showing that. Again, so having above 11 is really, really bad, and they're not managing their hyperglycemia very well. However, if a diabetic takes too much insulin and the insulin works too fast or too quickly and too much glucose is absorbed, it can cause them to go into a hypotonic state, kind of get low blood sugar or hypoglycemia, feel shaky and weird, and can also be caused from taking just pills or insulin related shots. And so if you listen to some of our guest speakers or you did the activity about life of a diabetic, you can see how those people with pumps constantly use their pumps to look at how their hypo or hyperglycemic areas or attacks are going on. And they do this by pricking their finger and checking their blood several times a day. And then if they have an insulin pump, it gives it a steady release throughout the day. Not all pumps will actually detect that for diabetics. They still have to prick their fingers. So that's the good news, is that we're having better treatments. We're able to diagnose diabetes earlier using things like the A1C test. Proactive equals a healthier life and a longer life. And we're doing lots of new research. However, we still know that there's no cure for diabetes. Here are some of the essential questions and key terms you should be familiar with at the end of this presentation and end of activities 2.3 to know for the final and the test. Make sure you take a look at these. Here are some terms defined for you when it comes to iso, hypo, and hypertonic, talking about cells and osmoregulation, or osmo meaning water regulation, how our body regulates that. Here's another little diagram to show you kind of animal cell versus a plant cell when it comes to swelling and shrinking. It doesn't matter if you're animal or plant, it still kind of affects the body the same way. Isotonic keeps the body at a normal, even rate cell-wise, whereas hypo causes them to swell, and they can actually pop or lyse, and hypertonic causes them to shrink, shrivel up, and that can actually cause the membrane to become all wilted and not burst, but break as well. After we're done with activity 2.3.2, where we're going to go from here is we're going to look at different complications that diabetes can have on the body. You're going to do some research. You're going to look at those effects, create a little graphic organizer for yourself, and make sure that you're, again, familiar with these key terms and essential questions when it comes to activity 2.3.